Understanding RNA Based Therapies, from antisense oligonucleotides to mRNA. Nucleic acid-based therapeutics represent a groundbreaking frontier in modern medicine, utilizing the precise targeting capabilities of synthetic or naturally occurring DNA and RNA molecules to modulate gene expression. These therapies, also referred to as oligonucleotide therapeutics or gene silencing technologies, interact directly with specific RNA or DNA sequences to influence cellular function, offering unprecedented opportunities to treat a wide range of diseases by either silencing harmful genes, enhancing beneficial ones, or modifying how genetic messages are processed. These molecules provide innovative solutions for genetic disorders, cancers, infectious diseases, and neurodegenerative conditions. As a cornerstone of personalized medicine, nucleic acid-based therapies hold the potential to address the root causes of diseases that were previously considered untreatable, shifting the focus from symptom management to precise genetic intervention. This emerging field encompasses a variety of therapeutic modalities, each with distinct mechanisms of action. Antisense oligonucleotides, ASOs, are designed to bind complementary RNA sequences, effectively blocking or altering protein production, while small interfering RNAs, siRNAs, utilize the RNA interference, ARNA, pathway to degrade specific mRNA targets with high precision. Other strategies, such as microRNAs, exploit naturally occurring gene-regulating molecules to fine-tune cellular processes. RNA aptamers, short RNA or DNA sequences with antibody-like binding properties, target proteins with exceptional specificity, while ribozymes function as catalytic RNA molecules to cleave specific RNA sequences. Meanwhile, mRNA therapeutics, exemplified by the widely used COVID-19 vaccines, enable cells to produce therapeutic proteins directly, opening new avenues in vaccines, cancer immunotherapy, and enzyme replacement therapy. Collectively, these approaches exemplify the power of nucleic acids as versatile tools in molecular medicine. While the potential of nucleic acid-based therapeutics is immense, their practical application comes with challenges. Delivery systems, such as lipid nanoparticles, are critical for protecting these molecules and ensuring their uptake by target cells. But achieving efficient and tissue-specific delivery remains an ongoing hurdle. Stability is another concern, as unmodified nucleic acids are prone to rapid degradation by enzymes in the body. Advances in chemical modifications, including the incorporation of modified nucleotides and protective structures, have significantly improved their stability and functionality. Additionally, minimizing off-target effects to ensure precision and reduce unintended interactions is a key area of research. Despite these obstacles, the field continues to advance rapidly, driven by innovations in molecular biology, chemistry, and nanotechnology. With these strides, nucleic acid-based therapeutics are poised to transform medicine by addressing the molecular underpinnings of disease and offering highly targeted, effective, and customizable treatments for a wide array of conditions. Nucleic acid-based therapeutics, also known as oligonucleotide therapeutics or gene silencing technologies, utilize synthetic or naturally occurring DNA or RNA molecules to modulate gene expression for therapeutic purposes. These molecules function by interacting with specific RNA or DNA sequences to either suppress, enhance, or alter gene activity. Key subcategories include antisense oligonucleotides, ASOs, which are synthetic single-stranded DNA or RNA molecules that bind to complementary RNA to block or modify gene expression. Small interfering RNAs, CSIs, which are double-stranded RNAs that silence target mRNAs via the RNA interference, SIS, pathway, and microRNAs, miRNAs, naturally occurring small RNAs that regulate gene expression by binding to mRNAs to inhibit translation or promote degradation. Other classes include RNA aptamers, short RNA or DNA sequences that fold into specific structures to bind and inhibit proteins like antibodies, and ribozymes, catalytic RNAs designed to cleave specific RNA sequences. Additionally, mRNA therapeutics involve synthetic mRNAs that direct the production of therapeutic proteins, as seen in mRNA vaccine. Antisense oligonucleotides, ASOs, are short, synthetic, single-stranded DNA or RNA molecules designed to bind specifically to RNA sequences via complementary base pairing, thereby modulating gene expression. Engineered to interfere with the normal function of RNA, ASOs are typically 12-25 nucleotides in length, optimized for both specificity and biological functionality to ensure their stability in the body. Where unmodified oligonucleotides would be rapidly degraded by nucleases, ASOs incorporate several chemical modifications. Common enhancements include a phosphorothioate backbone, in which one non-bridging oxygen in the phosphate group is replaced with sulfur to increase nucleus resistance, 2 modifications on the sugar ring, such as 2-O-methyl or 2-O-methoxyethyl, to improve stability and reduce off-telomere effects, and locked nucleic acids, LNAs, which constrain the sugar conformation to boost binding affinity. In addition, ASOs may be conjugated to delivery-enhancing molecules like lipids or peptides to improve cellular uptake and therapeutic efficiency. Antisense oligonucleotides, ASOs, exert their therapeutic effects by binding to specific RNA targets, with their mechanism of action falling into 3 main categories. First, RNA cleavage via RNase H occurs when the ASO forms a DNA-RNA hybrid with the target RNA, which is then recognized by the enzyme RNase H. This enzyme cleaves the RNA strand, effectively degrading the NAR and preventing protein translation. Second, steric hindrance involves the ASO binding to critical regions of the mRNA, such as the start codon or ribosome binding site, physically blocking the translation machinery and thus inhibiting protein synthesis without degrading the mRNA. Third, in splicing modulation, ASOs bind to pre-mRNA and interfere with normal splicing by masking splice sites or regulatory sequences, leading to altered splicing outcomes. This can result in exon skipping, inclusion of alternative exons, or changes in the reading frame, ultimately producing a modified or truncated protein. Antisense oligonucleotides, ASOs, are highly specific synthetic strands of DNA or RNA that bind to target RNA sequences to regulate gene expression. Therapeutically, ASOs are used in a range of diseases caused by faulty or overactive genes. In genetic disorders, such as spinal muscular atrophy, nusinersen, Spinraza, modifies SMN2 splicing to produce functional protein. In cancer, ASOs can silence oncogenes like BCL2, which supports tumor cell survival. For infectious diseases, ASOs can block viral RNA replication, while in neurodegenerative disorders like Huntington's disease, they reduce levels of toxic proteins by degrading mutant mRNA. ASOs also serve as powerful research tools, enabling targeted gene silencing in vitro and providing insights into RNA processing and splicing mechanisms. Their major advantages include high specificity, allowing selective binding to RNA targets, customizability, making nearly any RNA sequence addressable, including those considered undruggable, and broad applicability, suitable for correcting splicing defects, silencing harmful genes, or blocking toxic RNA. Challenges and limitations. While antisense oligonucleotides, ASOs, offer precision in gene silencing, they face key challenges. Delivery to specific tissues or cells remains a major hurdle. Approaches like lipid nanoparticles, cell-penetrating peptides, and GalNAc ligands for liver targeting are under development to improve uptake. Off-target effects due to imperfect base pairing can lead to unintended gene silencing, while stability and immunogenicity remain concerns. ASOs, despite chemical modifications, may still degrade prematurely or trigger immune responses. Additionally, cost is a limitation, as synthesis and large-scale production remain expensive. Future directions. Advances are focusing on enhanced delivery systems, such as exosome-based carriers and next-generation nanoparticles, and novel chemical modifications to improve pharmacokinetics and reduce side effects. ASOs also hold promise in personalized medicine, where they can be tailored to a patient's unique genetic mutations. Small interfering RNAs, cRNAs. siRNAs are short, double-stranded RNA molecules, typically 21 to 23 nucleotides, central to the RNA interference, RNA pathway, a conserved cellular mechanism for regulating gene expression. siRNAs can be synthetic, for experimental or therapeutic use, or endogenous, processed from longer RNA precursors. Their main function is to silence genes by guiding the degradation of complementary mRNA with high sequence specificity. Small interfering RNAs, cRNAs, are short, double-stranded RNA molecules, typically 19 to 21 base pairs in length, with 2 nucleotide 3Rs overhangs on each strand that enhance their recognition and loading by the RNA interference, RNA, machinery. Each cRNA consists of a guide antisense strand, which is complementary to the target RNA and directs gene silencing, and a passenger sense strand, which is usually degraded during processing. The mechanism of siRNA action involves several key steps within the RNAi pathway. First, siRNAs are delivered into the cytoplasm using methods such as lipid nanoparticles or chemical modifications that improve stability and uptake. Once inside the cell, the siRNA duplex is recognized and loaded into the RNA-induced silencing complex, RISC. Within RISC, the Argonaute-2 AGO2 protein selects the guide strand based on the thermodynamic properties of the duplex and discards the passenger strand. The RISC-CA-ORA complex then scans cellular mRNAs for complementary sequences. When a match is found, the guide strand binds to the target mRNA via Watson-Crick base pairing, allowing AGO2 to cleave the mRNA, leading to its degradation and effectively silencing gene expression. Once the ARNA guide strand binds to its complementary mRNA sequence, Argonaute-2 AGO2 within the RNA-induced silencing complex, RNA-ASC, cleaves the mRNA at that site, preventing its translation into protein. The cleaved mRNA is then degraded by cellular exonucleases. After cleavage, RISC is recycled and can target additional mRNA molecules, amplifying the silencing effect. RT-RNAs are valuable tools in both research and therapeutic applications due to their high specificity in targeting genes. In research, they are widely used for gene knockdown studies to investigate gene function and to dissect cellular pathways. Therapeutically, cRNAs are being developed to treat various conditions at the mRNA level. For example, patisiran Onpatro, the first FDA-approved siRNA drug, treats hereditary transthyretin-mediated amyloidosis by silencing the TTR gene. cRNAs are also being explored in cancer therapy to target oncogenes and drug resistance genes, in viral infections to block replication, in neurodegenerative disorders like Huntington's disease, and in ocular diseases such as age macular degeneration, by targeting angiogenesis genes like VEGF. Therapeutic application. cRNAs are increasingly being developed as precision medicines to treat diseases at the mRNA level by silencing specific gene transcripts. A prominent example is patisiran Onpatro, the first FDA-approved siRNA therapeutic used to treat hereditary transthyretin-mediating amyloidosis by targeting and silencing the gene encoding transthyretin, TT. In cancer, cRNAs can be designed to silence oncogenes, genes that inhibit tumor suppressors, or those involved in chemoresistance, offering a targeted approach to disrupt cancer progression. For viral infections, cRNAs inhibit replication by targeting viral RNA directly. or host cell factors essential for the viral life cycle. In neurodegenerative diseases like Huntington's and Alzheimer's, siRNAs are being investigated to reduce expression of genes contributing to the buildup of toxic proteins. Similarly, in ocular diseases, particularly age macular degeneration, siRNA therapies are being developed to target genes like VEGF involved in pathological blood vessel growth. Physical design considerations for designing effective siRNAs requires optimizing several factors to ensure specificity, potency, and stability. Sequence specificity is essential. Ideal siRNAs must perfectly match the target mRNA to reduce off-target effects. Target sites with complex secondary structures like hairpins are avoided to ensure effective binding. Thermodynamic asymmetry is also key. siRNA is designed with lower stability at the 5' end of the guide strand to promote correct strand selection by RISS. Chemical modifications improve therapeutic potential. 2,0-methyl or 2-fluoro-sugar modifications enhance nuclease resistance and reduce immune activation, while phosphorothioate linkages further stabilize the molecule. To prevent off-tangle effects, cRNA sequences are screened across the genome, especially the seed region, nucleotides 2,8, which plays a critical role in unintended interactions. Delivery remains a major hurdle. Lipid nanoparticles are commonly used for systemic delivery, particularly to the liver, while GalNAc, N-acetyl-galactosamine conjugation, targets siRNAs to hepatocytes via receptor-mediate uptake, improving precision and efficiency. siRNAs belong to a larger class of RNA-based therapeutics and differ in key ways from related technologies. Compared to antisense oligonucleotides, ASOs, which are single-stranded molecules that typically degrade RNA via RNase H or block translation through steric hindrance, siRNAs are double-stranded and operate through the RNA-induced silencing complex, RSC, which cleaves the target. RNAs tend to be more efficient due to the recycling capability of RISC, allowing one siRNA molecule to silence multiple targets. Though their delivery is more complex, compared to microRNAs, mRNAs, siRNAs are synthetic and precisely designed to target a single mRNA with perfect complementarity. Despite their promise, CMAs face several important limitations. Delivery barriers are a major challenge. Systemic administration often leads to CMAN accumulation in the liver, while delivery to other tissues such as the brain, muscle, or lung remains difficult. Off-target effects continue to be a concern, but improved design tools and sequence screening are reducing unintended interactions. Immune activation is another hurdle, as siRNAs can stimulate innate immune responses, including toll-like receptor signaling. However, this is being addressed through chemical modifications that reduce immunogenicity. Looking ahead, siRNAs are evolving beyond traditional applications. Expanding applications include emerging roles in gene editing, synthetic biology, and precision RNA tools, positioning siRNAs as versatile components of next-generation therapeutics. MicroRNAs, MION, is a strong energy signal that are detailed to spur nucleotides and motivate gene expression in eukaryotic cells and play crucial roles in development, cellular function, and disease. These small, non-coding RNA molecules typically range from 20 to 24 nucleotides in length and modulate gene expression at the post-transcriptional level. What are mRNA? Unlike cRNAs, which are synthetic and designed for perfect complementarity to a single mRNA, mRNAs are endogenous and naturally regulate gene expression by binding to partially complementary sequences in the 3 translated regions, UTRs, of target mRNAs. This interaction usually results in translational repression, which blocks protein synthesis, or mRNA degradation, which reduces transcript levels. Functionally, mRNAs act as gene expression dimmer switches, subtly adjusting protein output rather than completely turning it off. mRNA biogenesis, how are mRNAs made, is a multi-step process that occurs in both the nucleus and cytoplasm. It begins with the transcription of mRNA genes, which are embedded within the genome and transcribed by RNA polymerase II or III into long primary transcripts known as pri-mRNAs. These pri-mRNAs can span several kilobases and typically contain one or more hairpin structures, which serve as precursors to mature mRNAs. This intricate production pathway ensures precise regulation and integration of mRNAs into cellular gene control networks. Following transcription, microRNA, double-bind@mRNA, biogenesis, continues through several essential steps. In the nucleus, step 2 involves the processing of the primary mRNA, double-bind@mRNA, by the microprocessor complex, which includes the RNase 3 enzyme, DROCHA, and the RNA binding protein DGCR8. This complex recognizes and cleaves the hairpin structure of the primary mRNA to produce a tilde-70 nucleotide precursor, mRNA, pre-mRNA. Step 3 involves the nuclear export of the pre-mRNA by exportin 5 in a RAN-GTP-dependent manner, delivering it to the cytoplasm. In step 4, the pre-mRNA is further processed by DICER, another RNase 3 enzyme, which cleaves off the loop of the hairpin to generate a tilde-22 nucleotide mi. Finally, in step 5, one strand of the duplex, known as the guide strand, is incorporated into the RNA-induced silencing complex, RISC, where it associates with organoproteins, particularly AGO2, the catalytic component. The other strand, known as the passenger strand, is typically degraded. mRNAs regulate gene expression by guiding RISC to complementary sequences in the 3 untranslated regions, 3 UTRs, of target mRNAs. The functional outcome depends on the degree of base pairing between the mRNA and its target. In plants where pairing is often perfect or near perfect, the mRNA is cleaved by AGO2, leading to rapid degradation and complete suppression of protein production. In animals, where base pairing is generally imperfect, the result is usually translational repression, a block on protein synthesis, and/or mRNA destabilization, leading to gradual degradation. Through this mechanism, mRNAs act as fine-tuners of gene expression, adjusting protein levels without necessarily turning off gene function entirely. In animals, partial complementarity between a microRNA, miRNA, and its target mRNA is far more common than perfect pairing. In these cases, the miRNA RISC complex does not cleave the mRNA directly, but instead exerts its effect through 2 main mechanisms. Translation repression occurs when ribosomes are blocked from translating the mRNA into protein, effectively halting protein synthesis without degrading the mRNA immediately. Over time, the mRNA undergoes destabilization, which includes accelerated dedenylation, removal of the protective poly A tail, and decapping, followed by degradation by exonucleases. This layered regulation allows mRNAs to fine-tune gene expression gradually and reversibly, which is crucial for dynamic cellular processes. Targeting specificity is governed primarily by the seed sequence, a short, highly conserved region spanning nucleotides 2-7 at the 5 end of the mRNA. Perfect complementarity in this region is critical for effective binding to target mRNAs. However, targeting is also influenced by target site context, including the surrounding sequence, mRNA secondary structures, and accessibility. A single miRNA can regulate hundreds of different mRNAs, making them central nodes in gene regulatory networks. Moreover, cooperative regulation is possible when multiple miRNAs bind to different sites on the same mRNA, amplifying or refining the silencing effect. Because of this widespread regulatory capacity, mRNAs play crucial roles in development and differentiation, controlling cell fate decisions and tissue partnering. For example, miR-1 is a key regulator of muscle cell differentiation, highlighting the specificity and importance of individual miRNAs in shaping cellular identity. MicroRNAs, miRNAs, play essential roles in maintaining physiological balance and responding to disease. In homeostasis, miRNAs fine-tune critical cellular processes such as apoptosis, proliferation, and metabolism. For example, MIR3021 is known to regulate cell survival pathways under stress, contributing to cellular resilience. In the immune response, miRNAs such as miR145 play key roles in modulating inflammation and cytokine production, fine-tuning immune signaling. In cancer, miRNAs can act as either oncogenes, oncomias, or tumor suppressors. Oncomias, like miR21, promote cancer by down-regulating tumor suppressor genes, while tumor suppressive miRNAs, like the LET7 family, help prevent tumor development by silencing oncogene. mRNA dysregulation is also implicated in neurological disorders, including Alzheimer's and Parkinson's disease. For instance, MIR0124 is essential for neuronal differentiation, and its disruption has been linked to impaired neural function and neurodegeneration. Given their broad regulatory roles, therapeutic applications of miRNAs are an expanding area of biomedical research. miRNA are synthetic RNAs designed to restore the function of down-regulated or lost RNAs in diseases such as cancer, where reintroducing tumor-suppressive RNAs can help suppress oncogene expression. In contrast, anti-miRs, also called mRNA inhibitors, such as antagamirs or locked nucleic acids, LNAs, are engineered to inhibit overactive mRNAs that contribute to disease. One example being the inhibition of miR1022 to treat hepatitis C virus, HCV, infection. Beyond direct therapeutic use, miRNAs are also being developed as biomarkers for disease diagnosis and prognosis. Because mRNA expression patterns change in response to pathological conditions, circulating miRNAs in blood or other body fluids offer non-invasive indicators of disease states such as cancer, cardiovascular disorders, and neurological decline. Challenges in miRNA therapeutics remain a major hurdle for their clinical success. One of the most significant barriers is delivery, ensuring that miRNA mimics or inhibitors reach the correct tissues or cell types in sufficient concentrations. To overcome this, researchers are exploring lipid nanobodies, viral vectors, and conjugated oligonucleotides such as GalNAc, which facilitates liver-specific delivery. Another issue is off-tangle effects. Since mRNAs naturally regulate multiple genes, increasing the likelihood of unintended gene silencing and unwanted side effects. Additionally, stability poses a problem, as unmodified mRNAs are prone to rapid degradation by nucleases in the bloodstream. To address this, chemical modifications are often introduced to improve durability. Finally, immune activation is a concern. The body may recognize synthetic or exogenous miRNAs as foreign, triggering toll-like receptor TLR-mediated immune responses that can cause inflammation or other adverse reactions. Comparison with siRNAs highlights both functional similarities and key differences between these two classes of small regulatory RNAs. While both miRNAs and siRNAs regulate gene expression post-transcriptionally, they do so through distinct mechanisms. siRNAs are typically synthetic, double-stranded RNAs designed to bind with perfect complementarity to a single target mRNA, leading to direct cleavage by Argonaute-2 within the RNA-induced silencing complex, RISC. This results in potent, highly specific gene silencing. In contrast, mRNAs are endogenous, single-stranded RNAs that bind with partial complementarity to the three translated regions, 3 UTRs, of multiple mRNAs, usually leading to translation repression or mRNA destabilization, rather than cleavage. While cRNAs are better suited for precise, targeted gene knockdown, miRNAs function more broadly as regulators of gene networks and offer additional value as biomarkers for disease diagnosis and prognosis. RNA aptamers, a detailed exploration. RNA aptamers are short, single-stranded RNA or DNA molecules that fold into unique three-dimensional structures, enabling them to bind with high specificity and affinity to a wide range of targets, including proteins, small molecules, and even entire cells. Functionally similar to antibodies, aptamers offer distinct advantages due to their small size, synthetic production, and ease of chemical modification. These features make them attractive for therapeutic and diagnostic use, particularly in areas where antibody-based approaches may fall short. What are RNA aptamers? Aptamers function as molecular keys, engineered to fit specific locks in the form of target molecules. Their binding capabilities arise from complex secondary and tertiary structural elements, such as hairpins, loops, bulges, and pseudoknots, that allow them to form highly selective and stable interactions. These structures grant aptamers high binding affinity, strong interaction with the target, and specificity, selective targeting, qualities that are critical for precision applications in molecular medicine. How are RNA aptamers made? The most common method for generating RNA aptamers is a technique called SELEX, systematic evolution of ligands, by exponential enrichment. This process begins with random library generation, where a large pool of diverse single-stranded RNA sequences, typically 4,100 nucleotides long, is synthesized. These sequences include a randomized region flanked by constant primer binding sites. In the binding to target step, this RNA pool is incubated with a target molecule, commonly a protein, allowing sequences that bind to be separated from non-binders, which are washed away. Next comes amplification, where the bound RNA is reverse transcribed into complementary DNA, cDNA, amplified via PCR, and transcribed back into RNA to regenerate the pool. This is followed by iterative selection, where the binding, washing, and amplification steps are repeated over 15 rounds to enrich for high-affinity binders. Finally, in cloning and sequencing, the enriched aptamer pool is cloned and sequenced to identify the best candidates for further testing and development. How do RNA aptamers work? RNA aptamers exert their function by binding to their targets through precise non-covalent interactions, including hydrogen bonds, van der Waals forces, and electrostatic interactions. The key to their specificity lies in the aptamer's unique three-dimensional folding, which creates a surface that is structurally complementary to the target molecule. Much like a key fits into a lock, on binding, aptamers can inhibit the biological activity of their targets by blocking active sites or interaction domains, such as preventing an enzyme from catalyzing a reaction or a receptor from binding to its ligand. Additionally, aptamers can serve as molecular sensors, detecting the presence of their targets with high specificity and often being coupled with signaling systems to produce a measurable response. Apply applications of RNA aptamers. RNA aptamers have broad potential across multiple domains of research and therapeutics, thanks to their ability to bind specifically and tightly to biological targets. In therapeutics, aptamers are used as inhibitors of disease-associated proteins. A notable example is pegaptinib, Macugen, the first FDA-approved RNA aptamer which binds and inhibits vascular endothelial growth factor, AHD, to treat age macular degeneration, AMD. Aptamers are also emerging as tools for drug delivery, where they function as targeting ligands that guide therapeutic payloads, such as nanoparticles or small molecules, directly to specific cells, like tumor cells in cancer therapy. Additionally, aptamers are being developed as anticoagulants, capable of selectively inhibiting coagulation factors such as thrombin, with the advantage of being reversible, a major benefit over traditional blood thinners. Diagnostics. RNA aptamers also have powerful applications in diagnostics, thanks to their ability to bind specific biomarkers with high affinity and selectivity. They are commonly integrated into biosensors, where they serve as recognition elements for detecting disease-associated molecules. For example, aptamer-based sensors can detect small molecules like glucose or proteins such as cardiac troponin, a key biomarker for diagnosing heart attacks. These systems offer rapid, sensitive, and often label-free detection methods, making them promising tools for point-of-care diagnostics research tools. In addition to therapeutic and diagnostic use, RNA aptamers serve as valuable research tools. In protein characterization, aptamers can act as molecular handles to selectively isolate and study specific proteins, similar to how antibodies are used in immunoprecipitation assays. For imaging applications, aptamers can be labeled with fluorescent tags to visualize their targets in live cells or tissue samples, enabling high-resolution tracking of molecular interactions in real time. Advantages of RNA aptamers. RNA aptamers offer a number of compelling advantages over traditional antibodies. Their synthetic and chemical nature allows for consistent, reproducible production without the need for animal hosts or cell cultures, ensuring reliable quality across batches. Their small size, ~15 kDa compared to ~150 kDa for antibodies, enhances tissue penetration and target accessibility. Additionally, aptamers exhibit low immunogenicity, reducing the risk of adverse immune responses, and many feature reversible binding, which can be switched off by introducing complementary oligonucleotides. Their broad target range also enables them to bind a diverse array of molecules, from small ions and metabolites to large proteins, with high specificity and affinity. Challenges in using RNA aptamers. Despite these advantages, RNA aptamers face several challenges that limit their widespread clinical use. A major concern is stability, as unmodified RNA aptamers are prone to rapid degradation by nucleases in biological environments. This issue can be mitigated with chemical modifications, such as incorporating 2-fluoro or 2-methyl groups, which enhance nucleus resistance. Another hurdle is their short half-life in circulation. Unmodified aptamers are quickly cleared from the bloodstream, but pegylation, the attachment of polyethylene glycol chains, can significantly prolong their systemic presence. Efficient delivery remains difficult, especially when targeting tissues beyond the bloodstream or crossing cellular membranes. Lastly, the cost of developing aptamers, particularly through the labor-intensive Celex process and chemical synthesis, can be high, posing a barrier to large-scale or commercial applications. Future directions for RNA aptamers. The next generation of RNA aptamer technologies is focused on expanding their stability, functionality, and therapeutic reach. One promising avenue is the engineering of superstable aptamers through the incorporation of unnatural nucleotides, or locked nucleic acids, LNAs, which dramatically enhance resistance to enzymatic degradation and extend their half-life in vivo. Another emerging area is the targeting of intracellular proteins, which requires developing effective delivery systems to transport aptamers across cellular membranes, an essential step for accessing and modulating protein targets inside cells. Further innovation lies in the development of aptamer-based drug conjugates, where aptamers are linked to therapeutic agents, such as small molecule drugs or toxins. These conjugates act like guided missiles, delivering their payloads directly to diseased cells, particularly promising for targeted cancer therapy, where precision is critical to minimize damage to healthy tissue. Finally, a cutting-edge direction is the integration of aptamers with CRISPR technology. Aptamers can serve as programmable guides or regulators in CRISPR-based gene editing systems, enhancing targeting accuracy or acting as switchable control elements in synthetic biology circuits. These innovations highlight the growing versatility of RNA aptamers in both fundamental research and next-generation therapeutics. Ribozymes, a technical deep dive. Ribozymes, or RNA enzymes, are unique RNA molecules that possess catalytic activity, distinguishing them from most RNAs that primarily act as carriers of genetic information. These functional RNAs are capable of catalyzing specific biochemical reactions such as RNA cleavage, ligation, and even peptide bond formation. Ribozymes play essential roles in natural cellular processes, most notably in the ribosome, and are being explored for use in therapeutic and biotechnological applications due to their precision and programmability. What are ribozymes? A ribozyme is a single-stranded RNA that folds into a defined three-dimensional structure which enables it to catalyze a chemical reaction without the need for protein enzymes. This catalytic activity stems from the RNA's ability to form complex secondary and tertiary structures like stem loops and pseudoknots, which help position reactive groups precisely for catalysis. Additionally, ribozymes often coordinate metal ions, particularly Mg2+, which can stabilize the reaction's transition state or directly participate in catalysis. The key functions of ribozymes include the cleavage of phosphodiester bonds, breaking RNA strands at specific sites, as well as the ligation of RNA fragments to join pieces together. Remarkably, one of the most critical natural ribozymes is found in the ribosome, where it catalyzes peptide bond formation during protein synthesis. Ribozymes in nature. Ribozymes are not just laboratory curiosities. They occur naturally across all domains of life and play essential roles in RNA processing, gene regulation, and protein synthesis. One class, known as self-cleaving ribozymes, catalyzes their own excision from longer precursor RNAs. A well-known example is the hammerhead ribozyme, commonly found in viroids and satellite RNAs of plants, where it enables precise self-cleavage to process viral. Another major natural function of ribozymes is seen in the ribosome, where ribosomal RNA performs the critical catalytic function. In prokaryotes, the 23 srRNA, and in eukaryotes, the 28 srRNA, act as the peptidylary transferase center, catalyzing the formation of peptide bonds during protein synthesis, firmly establishing the ribosome itself as a. Additionally, RNA splicing is mediated by ribozymes in the form of group I and group II introns, which catalyze their own excision from precursor RNAs and ligate the remaining exons, functioning without the need for protein enzymes. How do ribozymes work? The catalytic mechanism of ribozymes is rooted in fundamental principles of organic chemistry, including acid-base catalysis, nucleophilic attack, and transition state stabilization. Their activity begins with RNA folding, in which the ribozyme adopts a specific and highly ordered three-dimensional shape. This structure is formed through a combination of secondary interactions, such as base pairing, and tertiary interactions like pseudoknots and metal-ion coordination, often involving Mg2+ ions, that stabilize folding and assist in catalysis. Following proper folding, substrate binding occurs. The ribozyme identifies its RNA target via Watson-Crick base pairing, or by recognizing the substrate's shape through structural complementarity, precisely positioning it for catalysis. This structural precision is essential for the ribozyme's efficiency and specificity, making it a highly evolved RNA catalyst. Cleavage reaction. The fundamental biochemical activity of many ribozymes is site-specific RNA cleavage, achieved through a well-orchestrated catalytic mechanism. The reaction typically begins with the activation of the 2-hydroxyl TOH group on the ribose sugar of the substrate RNA. This activation is facilitated either by the ribozyme structure or by a metal ion cofactor, most commonly magnesium, Mg2+, which acts as a general base to deprotonate the 2OH, converting it into a potent nucleophile. In the next step, this nucleophilic OH group attacks the adjacent phosphate group at the 3 position, leading to cleavage of the phosphodester bond. The result is the formation of a cyclic 2 3-OH phosphate at the cleavage site and a free 5-hydroxyl group on the other cleavage product. Hallmarks of ribozyme-mediated RNA cleavage. Types of ribozymes. Ribozymes are categorized into several classes based on their origin, catalytic function, and potential for engineering. Small self-cleaving ribozymes are compact, catalytic RNAs that typically perform single turnover cleavage reactions. Examples include the hammerhead ribozyme, found in plant viroids and satellite RNAs, the hepatitis delta virus, HDV ribozyme, essential for viral replication, and the hairpin ribozyme, which requires divalent cations such as Mg2+ to cleave RNA at specific sites. These small ribozymes are widely studied for their minimal structural requirements and catalytic precision. In contrast, large catalytic ribozymes, such as group I and group II introns, play more complex roles. Group I introns catalyze self-splicing using an external guanosine as a cofactor and are found in genes for rRNA, mRNA, and tRNA in certain eukaryotes and prokaryotes. Group II introns also self-splice, but form a lariat intermediate, similar to the eukaryotic spliceosome, suggesting an evolutionary link between RNA splicing mechanisms. There are also engineered ribozymes, which are created through in vitro selection techniques like CELEX to cleave or modify specific RNA sequences. These artificial ribozymes, including RNA-cleaving DNA-azymes and custom ribozymes, are being actively developed for therapeutic applications, where they can be tailored to silence disease genes by cleaving their RNAs with high specificity. Applications of ribozymes. Ribozymes have demonstrated wide-ranging potential across therapeutic, research, and synthetic biology domains. In therapeutics, they are being developed as gene-silencing agents, particularly for diseases driven by overexpressed or defective genes. Through targeted mRNA cleavage, ribozymes can be engineered to bind and cleave specific mRNAs, thereby reducing the production of harmful proteins. For example, ribozymes targeting viral RNAs in diseases like HIV or hepatitis B aim to suppress viral replication. In cancer therapy, ribozymes are being designed to degrade oncogenic mRNAs or those linked to drug resistance, while in neurological disorders, ribozymes may be used to cleave mutant RNA transcripts involved in Huntington's or Alzheimer's disease. In molecular biology, ribozymes serve as valuable tools for gene knockdown, enabling researchers to selectively inhibit gene expression and study gene function. They are also employed for RNA manipulation, helping scientists explore RNA structure, processing, and regulation. In synthetic biology, ribozymes are incorporated into genetic circuits to control gene activity dynamically. For instance, ribozymes can function as biosensors, detecting specific RNAs or chemicals, or act as toggle switches, regulating gene expression in response to environmental cues or molecular signals. Advantages of ribozymes. Ribozymes offer several key advantages. Their specificity allows them to be precisely tailored to target a single RNA sequence, greatly reducing the risk of off-tangle effects. They also exhibit catalytic efficiency, meaning a single ribozyme molecule can catalytically cleave multiple RNA substrates. True enzymatic behavior. Additionally, ribozymes benefit from synthetic accessibility. Unlike proteins, they can be readily synthesized and modified in vitro, making them versatile tools for both basic and applied sciences. Challenges of ribozymes. Despite their promise, ribozymes face a number of practical challenges. Stability is a major issue. Natural ribozymes are vulnerable to degradation by ribonucleases, RNases, in biological fluids. This can be mitigated with chemical modifications, such as incorporating 2-fluoronucleotides or locked nucleic acids, LNAs, to enhance nucleus resistance. Delivery is another hurdle. Getting ribozymes into specific tissues or cells efficiently remains difficult. Potential solutions include viral vectors, lipid nanoparticles, or aptamer conjugation. to facilitate targeted delivery. Lastly, ribozymes require accessible single-stranded regions within target RNAs to function, limiting their applicability to structured RNAs where such regions may be scarce or inaccessible. Examples of ribozyme-based therapeutics. Several ribozymes have been developed and tested in clinical and preclinical settings to demonstrate the feasibility of RNA-based catalytic gene silencing. Angiozyme is a ribozyme targeting VEGF receptor mRNA designed to inhibit angiogenesis and investigated for its potential in anti-cancer therapy. Heptazyme was developed to target hepatitis C virus, HCV RNA, aiming to block viral replication. Another notable example is TATREZ, a ribozyme engineered to cleave HIV TAT transcripts, thereby reducing viral gene expression and replication. These examples illustrate the diverse potential of ribozymes in combating viral infections and cancer through targeted RNA cleavage. Future directions. The future of ribozyme technology lies in expanding their functional repertoire and improving delivery and stability. One exciting area is the integration of ribozymes with CRISPR systems, where ribozymes can be used to process guide RNAs or regulate CRISPR activity at the RNA level, enabling programmable RNA manipulation. Advances in synthetic ribozyme engineering aim to create novel catalytic activities beyond traditional RNA cleavage, potentially allowing ribozymes to catalyze reactions such as ligation or chemical modifications. The combination of ribozymes with nanotechnology, such as nanopartisols or liposomes, may enhance delivery efficiency and protect ribozymes from degradation in vivo. These innovations open the door for more robust and customizable RNA tools in therapeutics and synthetic biology. mRNA therapeutics, a comprehensive exploration. Shifting to another transformative RNA modality, mRNA therapeutics enable the in vivo production of therapeutic proteins by delivering synthetic messenger RNA into cells. Once inside, the mRNA is translated by the host cell machinery into a functional protein. This approach is highly adaptable and has already demonstrated major clinical success, most notably with mRNA vaccines for COVID-19. Beyond infectious diseases, mRNA therapeutics are being developed for enzyme replacement therapy, genetic disorders, and cancer immunotherapy, offering a flexible platform that bypasses the need to deliver or alter DNA, while avoiding the risk of genomic integration. mRNA therapeutics involve the use of synthetic, chemically modified messenger RNA molecules to instruct a patient's cells to produce specific proteins that exert a therapeutic effect. The mechanism is straightforward. Once introduced into target cells, the synthetic mRNA is translated by ribosomes into a functional protein. Whether it's an antigen for vaccination, an enzyme for metabolic correction, or a signaling molecule for immune modulation, this platform offers several key advantages. First, it is non-permanent. Unlike DNA-based gene therapies, mRNA does not integrate into the genome, significantly lowering the risk of long-term genetic disruptions. Second, it allows for rapid development. Synthetic mRNA sequences can be designed and manufactured within weeks, making them especially valuable for responding to emerging pathogens like SARS-CoV-2. Lastly, the versatility of the technology means that virtually any protein, whether therapeutic, structural, or immunogenic, can be encoded, making mRNA therapeutics a universal platform for treating a wide range of diseases. How mRNA therapeutics are made is a carefully controlled, multi-step process designed to maximize their safety, stability, and translational efficiency. It begins with template design, where a DNA template is engineered to include all necessary regulatory and structural elements. A cap structure at the 5' end to enhance stability, and ribosome recruitment. A 5' untranslated region, UTR, optimized to regulate translation efficiency. The coding sequence, CDS, that determines the amino acid sequence of the therapeutic protein. A 318 longevity and translation rates. And finally, a poly-A tail, which protects the mRNA from enzymatic degradation and further boosts translational performance. Once the template is complete, in vitro transcription, IVT, is performed using bacteriophage-divided RNA polymerases, such as T7 or SP6, which generate linear synthetic mRNA from the DNA blueprint. This IVT step is typically optimized to incorporate chemical modifications, such as pseudouridine or 5-methylcytidine, that increase mRNA stability and reduce activation of innate immune sensors like toll-like receptors. Modifications are essential in the production of mRNA therapeutics to enhance their stability, reduce immunogenicity, and ensure efficient translation inside human cells. One key element is the incorporation of cap analogues, synthetic structures added at the 5' end of the mRNA, either enzymatically or co-transcriptionally, which mimic the natural mRNA cap and are critical for efficient ribosome recruitment. Another crucial component is the use of modified nucleosides, for example, replacing uridine with N1-methyl pseudouridine, which helps the mRNA evade innate immune detection and increases its stability in the cell. Additionally, poly-A tail optimization plays a vital role. The tail, typically consisting of 150 adenosines, is carefully designed to strike a balance between stability and translational efficiency. Purification follows synthesis and modification, and it is a critical phase in ensuring the quality and safety of the final mRNA product. During this step, techniques such as high-performance liquid chromatography, HPLC, or cellulose-based affinity purification are used to remove impurities, particularly double-stranded RNA. These dsRNAs are problematic because they can trigger strong innate immune responses, leading to inflammation or reduced therapeutic efficacy. Purification ensures that only the functional single-stranded mRNA is retained for therapeutic use. Delivery vehicle development addresses one of the core challenges of mRNA therapy: getting the fragile mRNA molecule safely into cells. Naked mRNA is rapidly degraded in the body and cannot cross cell membranes efficiently. To overcome this, the mRNA is encapsulated in delivery vehicles, with lipid nanoparticles, LNPs, being the most widely used and clinically validated system. These nanoparticles protect the mRNA from degradation, facilitate cellular uptake, and help release the mRNA into the cytoplasm where translation occurs. This step is especially critical, as the choice and design of the delivery system directly influence biodistribution, targeting efficiency, and immune tolerance. The mechanism of action in mRNA therapeutics involves a multi-step cellular process that enables synthetic mRNA to be translated into a therapeutic protein. Step 1. Delivery begins with the mRNA being packaged in lipid nanoparticles, LNPs, or similar carriers. These structures serve two critical functions. They protect the mRNA from enzymatic degradation by nucleases in the bloodstream and facilitate cellular uptake through endocytosis, where the cell engulfs. Once inside the cell, step 2, endosomal escape occurs. The LNPs are trapped in endosomes, but their lipid components are specifically engineered to destabilize the endosomal membrane, allowing the mRNA to escape into the cytoplasm, the site of protein synthesis. In step 3, translation, the freed mRNA is recognized by ribosomes, which translate its coding sequence into the desired therapeutic protein. This protein can serve multiple roles depending on the application. For instance, acting as a vaccine antigen, like the spike protein in COVID-19 vaccines, or replacing a faulty protein in a genetic disorder. Finally, step 4. Degradation ensures that the synthetic mRNA does not linger indefinitely. It is naturally broken down by cellular RNA turnover mechanisms, eliminating the risk of long-term persistence or genomic integration. Applications of mRNA therapeutics span a broad spectrum of medicine. One of the most successful areas is mRNA vaccines, which work by encoding antigens such as viral proteins that are translated inside the host cell and presented on the cell's surface to stimulate an immune response. The most prominent examples are the Pfizer BioNTech and Moderna COVID-19 vaccines, which encode the SARS-CoV-2 spike protein, triggering both antibody and T-cell responses. Another major application is protein replacement therapy, where mRNA is used to restore functional protein expression in patients with genetic diseases. For example, in cystic fibrosis, mRNA can encode a healthy CFTR protein, compensating for the defective gene. In cancer immunotherapy, mRNA is engineered to encode tumor-specific antigens, or immune modulators, helping the immune system recognize and attack cancer cells. For instance, mRNA vaccines may encode neoantigens, which are unique mutated proteins found only in tumors. In regenerative medicine, mRNA is being developed to promote tissue repair by encoding growth factors, or structural proteins to support healing in damaged tissues. Finally, in the field of infectious disease, mRNA technology is expanding beyond COVID-19 to target pathogens like influenza, HIV, Zika, and rabies, providing a flexible and rapid response platform for future vaccine development. The advantages of mRNA therapeutics are numerous and make this platform one of the most promising innovations in modern medicine. One of the most striking benefits is rapid development. As demonstrated during the COVID-19 pandemic, mRNA vaccines were designed and advanced to clinical use in record time, thanks to the flexibility and modularity of the platform. Another major advantage is the lack of genomic integration. Unlike DNA-based therapies that risk insertional mutagenesis, mRNA operates in the cytoplasm and does not integrate into the genome, eliminating concerns about altering host DNA. The platform is also highly customizable, allowing scientists to design mRNA sequences that encode virtually any therapeutic protein, which opens the door for personalized medicine and precision-surrogated treatment. Additionally, scalable manufacturing is a key strength. Synthetic mRNA can be produced rapidly in vitro without the need for live cells, making it ideal for mass production in emergency situations. Challenges of mRNA therapeutics, however, still need to be addressed to fully realize the platform's potential. Delivery remains one of the most critical issues. While lipid nanoparticles, LNPs, have been successful in enabling mRNA delivery into cells, targeting specific tissues, such as the brain or heart, is still a significant hurdle. Another limitation is stability. Unmodified mRNA is fragile and rapidly degraded by RNases in biological environments. This challenge is being mitigated through the use of modified nucleosides, e.g., pseudouridine, and stringent cold chain storage. But room temperature stable formulations are still in development. Immunogenicity also poses a concern. Synthetic mRNA can activate innate immune sensors like toll-like receptors, potentially leading to inflammation. This issue is being addressed by removing double-stranded RNA, C-Des-LR, DSLA, contaminants during purification and modifying mRNA chemically to reduce its visibility to immune surveillance. Lastly, cost remains a barrier, as mRNA therapeutics require expensive synthesis, purification, and cold storage, making global access and distribution more difficult. Future directions for mRNA therapeutics are already underway, promising to overcome current limitations and expand clinical applications. One exciting innovation is self-amplifying mRNA, SALRAN, which includes sequences encoding an RNA-dependent RNA polymerase, REDRP. This allows the mRNA to replicate itself inside cells, significantly reducing the required dose and improving cost efficiency. Efforts in targeted delivery are advancing, particularly through GalNAc-conjugated nanoparticles that enable precise delivery to the liver, with ongoing research into carriers for other tissues like muscle and the central nervous system. Work is also being done to develop thermostable formulations that can maintain mRNA integrity at room temperature, easing distribution and storage challenges in global health settings. Finally, the scope of mRNA therapeutics is rapidly expanding, with research targeting rare diseases, autoimmune conditions, and neurodegenerative disorders. positioning mRNA as a versatile tool across virtually every therapeutic area. Conclusion: nucleic acid-based therapeutics have ushered in a transformative era in medicine, redefining how we approach disease treatment by targeting its genetic roots. These advanced therapies leverage the unique properties of DNA and RNA to modulate gene expression with unparalleled precision, offering new hope for conditions ranging from rare genetic disorders to widespread challenges like cancer and infectious diseases. As the field continues to mature, these therapeutics promise not only to enhance patient outcomes, but also to reshape our broader understanding of disease management, shifting the focus from generalized approaches to highly tailored, molecular-level interventions. The future of this technology is marked by incredible potential. Emerging innovations in delivery systems, such as lipid nanoparticles and tissue-specific targeting strategies, are addressing long-standing challenges of stability and precision, while chemical modifications are further enhancing the safety and efficacy of nucleic acid molecules. Advances in mRNA therapeutics, already demonstrated in the success of COVID-19 vaccines, are paving the way for rapid responses to emerging diseases and new applications in cancer immunotherapy and protein replacement therapies. Meanwhile, the rise of personalized medicine, fueled by breakthroughs in genomic sequencing, is enabling the development of therapies tailored to the unique genetic makeup of individual patients. This synergy between cutting-edge molecular biology and clinical innovation heralds a future where treatments are not only more effective, but also more inclusive, addressing a broader spectrum of diseases.